Hi, in this video, we'll talk about SWI SNF nucleosome remodeling complex and how does it work and what its implication in health and diseases. So SWI SNF stands for switch and sucrose non-fermentable. This nucleosome remodeling complex was discovered in yeast in a genetic screen, which was not aimed to identify this kind of nucleosome remodeling complex, but it was aimed to identify mutants which are defective in mating type switching or sucrose fermenting capability. And thereby, mating type switching stands for sweet, SWI, and the sucrose non fermenting stands for SNF. And a few years later, this complex was purified. Now, the purified SWI SNF complex, when added to a chromatin in vitro, it has been seen that it can change the configuration of the chromatin and it can change the compaction and accessibility of this chromatin. That leads to an indication that this kind of complex might be super important in terms of changing the chromatin architecture. Now we know that this particular complex is super important in yeast for mating type switching. In mating type switching, there are two mating types. One is alpha, one is A. And this mating type switching requires a specific endonuclease known as HO endonuclease. Now, HO endonuclease is a protein, so it would be transcribed by a gene which is called the HO gene. Now, the HO gene body has several nucleosomes, and the DNA which is coding for this HO protein would be wrapped around histones or histone cores. So something need to loosen these DNA in order to get the access to the gene body and get it transcribed. Only when it is transcribed and translated, then the HO protein would be produced and the mating type switching can happen. In this context, SWI SNF nucleosome remodeling complex takes important role, and let us review that what type of role does it play. So imagine this is the HO gene body, and the red marking here denotes the transcription start site. Now the polymerase cannot get the access to the transcription start site because that portion is tightly wrapped around the histone core. It is totally inaccessible. Now this SWI SNF complex is recruited near that region, and it slides the nucleosome such that the transcription start site is now free and accessible to the RNA polymerase. And once it is accessible, it can now move along the gene body and transcribing the HO gene. Now, let us review the mechanisms by which the SWI SNF complex can work. The first thing the SWI SNF complex can do is change the nucleosome spacing. So the internucleosomal space has very deep implication in terms of gene expression. Let's say after sliding the histone cores in a certain manner, certain regions are now accessible to transcription factors or let's say polymerases. So that give rise to change in gene expression profile. Other than that, the SWI SNF complex can lead to eviction of histone in a certain region. As a result of histone eviction from this particular region shown here, there could be an accessible site where a transcription factor or a RNA polymerase can bind, and this lead to change in gene expression profile. Now, SWI SNF complex can also replace histone in a nucleosome to a histone variant, which might has implication in terms of packing and ultimately is important for gene expression. Now, in human, in yeast, in Drosophila, this SWI SNF complex, its structure, its function is pretty much conserved. Now let us look at the domain structure of the SWI SNF complex in order to understand its functionality. So it has its ATPase domain, which is so important. The ATPase domain hydrolyzes ATP, and these big complexes, which are at least having 8 to 11 subunits, requires ATP hydrolysis in order to perform a change at a chromatin level. Now, changing a chromatin landscape is not easy. It requires a lot of energy, and thermodynamically it is pretty complicated. Other than that, this nucleosome remodeling complex has a bromo or a chromo domain, which make it capable to recognize acetylated or methylated histones. Now, in human, this SWI SNF complex is known as BAF complex or PBAF complex. In BAF, BRM or BRG1 is the ATPase domain, whereas bromo domain is also present in this particular BAF complex. HSA is a domain which allows this BAF complex to interact with actin. BAF 155 and BAF 170 regulates the stability of this BAF complex and has implication in gene expression. Now, BAF complex has different type of functions. It is involved in dendrogenesis and modulating spine density. It was found to be involved in learning and memory. Now, BAF complex help to maintain the neuronal progenitors at a stem state. Now, other than that, it also regulates the proliferation versus differentiation balance in early brain development stages. Not only that, in many cancers like breast cancer, colon cancer, the gene mutations which are frequently occurred involves the BAF complex genes. Now, the genes which give rise to BAF complex are heavily mutated in several cancers. This implies that the functionality of BAF complex and BAF complex mediated nucleosome remodeling or changing in chromatin structure is very important in terms of development and diseases. So I hope you enjoyed this video. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.